3D printing club, and he was just printing molecules for fun. And then he found something out about it, and so he's going to share with us his presentation. It's called Printing Proteins, a Powerful and Pedagogical Practice.
Okay, cool. Thank you. Can you hear me okay?
Yes.
Okay, cool. So I was asked to start with a powerful and impacting quote. So I'll ask you all, if you had to pass on all the knowledge of humankind that we have presently to the future generations, and you only had one sentence, what would you say? Nobody's gonna shout anything?
42.
Figure it out.
So, the great American physicist Richard Feynman, which some of you may have heard about, and was also a very great science lecturer, he said that if we had to do that, the one sentence that he would pass on would be that everything is made of atoms. Tiny little wiggling points of matter that comprise everything. And in a sense, what I'm doing here is showing people what things are made of. Because what I've been doing, and I started doing this just for fun, because I did a lot of molecular biology work, is printing models of molecules, models of atoms. And in fact, in another quote from Feynman, in a discussion that he had with a friend who was a poet and artist, he was accused as a scientist of making things dull and ugly by piercing them apart. And his friend asked him, what do you see in a flower? And you scientists make it dull, you take out all the beauty from it. And he replied, well, I see much more than you do, because I can imagine all the organelles in the flower, all the molecular processes, every single thing that's going on. So what I would like to do in the next few minutes is just to give you a sense of what is going on, a sense of what... I want to show you the unseen, and more than that, I want to make it tangible, which is why I've been printing these things, rather than only showing them on the screen. So, how do I pass next slide? Oh, cool. Okay. So when I came to Brandeis last August, I had never seen a 3D printer before. To me, it was like sci-fi movie things. And when I went through the list of clubs, and I saw that there was a 3D printing club, I was like, oh, I gotta join that club. So during the club fair, I walked right up to Noah. How do I shoot lasers? Well, anyway, I walked right up to Noah, who was then the president, said, I want to join your club, and I want to print molecules. And I started doing that just for fun. Now, I want to be clear on one thing. 3D printing is not a new technology. 3D printing has been around for 30 years. But the reason why it exploded in the last 5 years or so is because the patents expired. And when the patents expired, all the open source community was able to start tinkering around and messing around and make things cheap and efficient without fear of being sued. And you're going to see yet more explosions of other 3D printing technologies, because the one patent that expired was for FDM technology, for extrusion-based printers. And in the next years, we are going to see a few others patents falling down, and this snowball is only going to get bigger. Anyway, that was my disclaimer. And the one technology that we use is fused deposition modeling, but it's really nothing more than just a glorified hot glue gun. What it does is it just lays tracks of extruded filament layer by layer, and then it lowers down the build plate and lays another layer and lowers again. So it's basically 2D printing over and over. Okay, having said all that, oh yeah, that's how it works. You just lay a track of filament, and then another track, and then you lower build plate and such. Okay, cool. So back to molecules and atoms. This is a vision that I believe at least the first one most of you should be familiar with. This is DNA. And here you see 4 different ways that you can represent a piece of DNA. The first one is actually the original figure that was published by Watson and Crick in the 1953 paper, in which they deciphered the structure of DNA and the secret of life and all that. And that's very nice, it's very pretty, it's the vision that we all have of when we think of DNA. And it's a diagram, it's good for conveying an immediate sense of shape, of connectivity, but it does not go on into the nitty-gritty details. The figure that you see next to it is a licorice or sticks representation, and it shows you every atom and how they are connected in space. And that's great, but it contains all the information, but it can be a bit daunting if you have a bigger molecule. Sometimes pieces of it can be obscured. The next one is also a vision that you might be familiar with. It's spheres or van der Waals representation. And atoms are not really solid spheres. You might be better off thinking of them as, soft fuzzy balls that can overlap with each other. But they do have a volume, and it's really hard to penetrate an atom beyond a certain length. And this is what this kind of representation conveys. It conveys more or less the volume occupied by each atom, beyond which you can't really push in. So if you look at this one, you would think, oh, there's a lot of empty space there. There's not really a lot of empty space that you can get off on without pushing things aside. And the last figure is, if you imagine yourself as a molecule, this is probably what you would feel. Because, if you're, say you're a water molecule, you have an oxygen and 2 hydrogen, and that's kind of big. So you can just kind of probe, the raw features of whatever other molecule you're looking at. So I have printed out this model of DNA that you see there as a surface. And we've been using this in Professor Daniel Crummell class. And I can't emphasize enough how important it is to be actually able to handle these things rather than just rotate them in the screen. And I'll pass this around to the audience. And don't worry, you cannot break that down unless you try really, really hard. Another word on the importance of models. These are Watson and Crick with their original DNA model. And this is Linus Pauling with his protein models. And in the 50s and 60s and 70s, all those great scientists who were cracking down the structure of matter, they were model building. They couldn't just keep to, they didn't have computers to draw things and visualize them as easily. And even if they had, there's quite nothing that can compare to, handling things around and messing with them and tinkering and trying to build a spatial understanding of how things are arranged. And a funny thing about Linus Pauling is that a few months before Watson and Crick published the double helix, Linus Pauling had actually published a triple helix model of the DNA, which was not quite right, but is one of the best scientific anecdotes of how you try and fail, and then you try again and get it right. Somebody else gets the credit.
Quantum mechanics, right? We're going out?
Sorry?
Quantum mechanics.
Yeah, that too. But there's a lot more feeling there. So, these guys were pioneers. Pauling and Watson and Crick. They had to do everything by hand. The way they figure out the structure of a molecule was you shoot X-rays at them because they're smaller than light, so you can't see them. You basically shoot X-rays at them and you see how the X-rays diffract. You see the shadow that is cast by whatever molecule you're looking at from several angles, and then you try to reconstruct the shape of the molecule from that. Which is why DNA was such a huge thing, because it's a quite complex molecule, and to do that by hand was not easy. But with computers and legions and legions of scientists nowadays, we have been able to crack down the atomic structure of matter at a really impressive pace. And this is just a sample of what they have done, and you can't quite see it, but take my word that these little dots are atoms. This is, I took this from the protein data bank. Every time scientists crack down a structure, they publish it, they get all the credit and fame, and they make it available for anyone else to download it and mess with it and use it for their studies. And right now they have over 100,000 structures, just like this, so much more complex. This is a virus, for example. Here is a cell membrane with stuff sticking through it. Here's DNA with all sorts of things that bind to it. And this is beautiful and this is daunting. And it's really hard sometimes to convey what's going on. You have to think really hard not to just present, a big blob of sticks and balls, and rather to try to show what really matters here. Because there's a lot of information here. And it can be also beautiful. It can be just beautiful. When I found this for the first time, I was like, "Oh, this is awesome!" And I just downloaded random structures and kept looking at them in any molecular visualization software that you may have, just because they were pretty. I didn't really care for the biology. So what I'll do in the last few minutes is I'll walk you through some of these really cool structures, just to give you a sense of how awesome things can be. And they are all made of atoms, and how can atoms make up all the stuff that we have around us. And I hope that some of you will get excited with this, and maybe when you go home you go to pdb.org and start checking this stuff out. So the first one that I have here is hemoglobin. You probably know that it's the guy that carries oxygen in your heme cells. And the way it does it is it has this little complex here with an iron. That's why you need iron too. That binds to oxygen, and when it binds to oxygen, it changes shape. And by changing shape, it actually has 4 heme complexes. It makes it more favorable for other oxygens to bind. And in my printed model, the light here is horrible, but take my word for it, there is a heme complex there. And here is that same model. Note that hemoglobin is made of 4 chains. So there are 4 heme groups here, and you can try to find them. Cool. Next molecule. Oh, this one is actually the blue thing is DNA, and the pink thing is a ribon diagram of a protein. Rather than showing you all the atoms, I'm just showing you the overall features. And what this guy is, this guy's called zinc finger. It's called zinc finger because it has these green zinc atoms that hold everything together because they have a positive charge. And what's cool about it is that you see that it's going into the DNA grooves. And it recognizes specific sequences of DNA. So with these 3 zinc fingers, each one recognizes 3 base pairs. You can actually recognize a sequence of 9 base pairs. And you can make machinery to go on and cut a very specific sequence of your DNA. And that's how you can do, for example, genetic engineering. You want to cut just that gene, or you want to insert something just there. You got to recognize that. And that's one way you can do it. So here I have the zinc finger. And I just want to show to everybody, because I was really surprised and proud of this print. This was not something very trivial to print with this technology, and it came out very nicely. So have fun, find the zinc atoms, and yeah, keep in mind that the ball thing is DNA, and this squiggly thing is protein. Oh, okay. This is a piece of RNA. You all know that we have DNA and we have the close cousin, that's RNA, and the only difference is that and the sugar that's after the base, they have or have not in oxygen. But you don't care about this. What you care is that RNA doesn't come in double strands, and so it's much less stable. But it can fold around itself and do a lot of cool stuff. And what you see here is actually just a single strand of RNA that's squiggled around and folded around itself in this funny shape. And it's so important. I choose this one. I really like this one because every single biologist knows what I'm talking about.
The amino acids are the
Oh, there's sound? How do I turn off sound? Okay, cool. Yeah, I want to do narration. Oh, yeah. So you see there are a lot of tRNAs floating, and oh, this video is going to take forever. It was supposed to be just a minute. Anyway, you see a lot of tRNAs floating, and here in the tip, what they do is they carry an amino acid, and they're called transfer RNA because they transport that amino acid to the ribosome, which is the machinery in the cell, the big protein assembly in the cell that synthesizes protein. They fit in there, and eventually the video is going to show that. They fit in there and they get recognized. And yeah, so this is the ribosome. Here is a message that's going in telling which amino acids you want. And here. So they come in just like a molecular factory, a production line, one after the other, let the RNA in and go out. Yeah, the video is really bad. But you get the idea. And then proteins are made with the red thing. Anyway. Oh yeah, and here is it. Sorry. This is actually one of my favorites. This is a nucleosome. So you all saw the little piece of DNA that we had. This is how it's actually packed in your cells. Because if you just extended your DNA in a straight line, it would go to the sun and moon, from here to the sun 8 times, back and forth. Or was it to the moon? I don't quite remember. but it was very large. So how do cells compact all that stuff? The way they do it is they have these special proteins, these orange guys there, they're called keystones, and around which the DNA wraps and forms like a string of beds. And this string of beds then wraps around itself again, like forming a telephone coil. And it wraps around again. Okay, so if this one does not like, I'll just let it run and it will speak for itself. But it wraps around itself again, like telephone coil, and then wraps around again and form your chromosomes. So there's a DNA, in comes a histone, another histone protein, they form a tetramer. There you go, string of beds. And this string of beds assemble around itself. Yep, telephone coil. There we go. This is called 30 nanometer fiber. You don't need to know that. There you go. The fiber weird. Yeah. You don't need me to tell this. It's cool stuff. And before you know it, chromosomes. So there you go. Oh, and by the way, because you have a continuous strand of DNA in principle, your chromosomes are made of a single molecule of DNA, supposing there are no breaks, which is pretty amazing. So, yep, just be careful not to break the DNA with that one. Okay, and our last guy, and one that I like a lot, this is a sodium channel. It's the guy that is responsible for your neurons being able to fire action potential. So when it's triggered, it opens and lets sodium ions into your neurons, and it sits in your cell membranes just like that. This stuff that you're seeing here is actually a real simulation. It's not an animation. It's not rendering anything. This is how the real thing probably looks like. This is like 20 seconds of sodium channel floating in in your cell membrane. And this is really cool because they can actually simulate the exact atomic motion of...
2 minutes in Roto, sorry.
don't need 2 minutes. But the one thing that I want to talk about the sodium channel is that it's made of 4 identical proteins that assemble together and form the channel. And when I printed it just like that picture that you see there. But I was very happy last year showing it to everybody and every professor, and everybody loved it. And one day I dropped it. And it's split in 2. But it's split in 2 right where the subunits meet. So that was actually really cool, because this is a nature-made molecular puzzle that you can all put together and see the hole where the sodium ions go through. So I'll disassemble it, and I challenge you to put it together because it's not so trivial. All right. So a few things where thanks is due. Thanks to the student union for throwing a lot of money at us. Like that's quite remarkable. It's not a trivial thing and should not be overlooked. The fact that they have thrown a few $1,000s in the club and made all this possible. Thank you to the Maker Lab, which is a space in the library put for by the LTS, and especially by Ian Roy, the bald guy over there. He's the real driving force behind all this. And I invite you, everybody, to go check out the Maker Lab anytime you want. From 7 to 9 every day, we have people there from the club. Oh, it's in the library above Starbucks. Yep. Thank you to the 3D Printing Club, but that's, I don't know, conflict of interest because I'm the president. And thank you for all the amazing people that we had there making this happen. And thank you all for tolerating me.